Hey, welcome back to Not So Complicated, and today we'll be talking about important pathological structures. All right, so out of 54, we'll be talking about 18 today, starting from Negri bodies, and these bodies are seen in patients suffering from rabies. All right, so this is how it looks like, and they are actually cytoplasmic eosinophilic inclusions in the pyramidal neurons of hippocampus, all right, and also in cerebellar Parkinson fibers. So the second is we have Lewy bodies, and these are particularly present in Parkinson's disease, and they look like that. And there are also irregular clumps of alpha-synuclein in substantia nigra neurons, all right. So and also cognitive function associated cells. So the third one we have is peak bodies, and they are like typically present in frontotemporal dementia, and they look like this. The structures are spherical, intracytoplasmic, misfolded tau protein inclusions. All right. So the fourth one we have is corpora amylacea, and they are present in aging brain, and they look very distinct in microscope and under the microscope, and these are small spherical polyglucosan bodies generated by astrocytes. All right, so these astrocytes are the macrophages in our brain. And also the fifth one we have is neurofibrillary tangles. All right, so they are commonly present in Alzheimer's disease and looks like that. These are abnormal clumps of hyperphosphorylated tau proteins, again tau proteins that form inside neurons. These structures are inside neurons, not in the glial region. All right, so the sixth one we have is Cowdrey type A inclusion. So these are present in herpes virus infection, HSV, herpes simplex virus, or herpes zoster virus. All right, so this is how they look like, and they are intranuclear inclusions with an eosinophilic core that has a clear halo around it. Remember this point, that has a clear halo around it. All right, so we have Cowdrey type B inclusion. They are present in cytomegalovirus and poliovirus infection, and they look like this, and they don't have clear halo around it. Otherwise it is very similar to Cowdrey type A. So intranuclear inclusions with an eosinophilic core that without a halo and is like circumscribed and multiple. All right, so we will be talking about Aschoff bodies next, and that are commonly present in rheumatic fever, and this is how they look like. This is a perivascular aggregation of large cells with polymorphic nuclei and basophilic cytoplasm, often arranged in a rosette shape around a central enucleated core of fibrinoid material. All right, so this is how Aschoff bodies looks like, and this is how you will write in the exams if it is asked in rheumatic fever. So and its cause cells, this is also seen in rheumatic fever, and this is also a content of Aschoff bodies. So they look like a caterpillar. All right, so a caterpillar like nucleus with a wavy linear condensation of chromatin, and in cross section the nucleus can appear wool-eyed. All right, so this is how anti-scar cells look like, and many people get confused with like anti-scar or anit-scar. It is anit-scar. It is a name of a scientist. All right, so that is how you should not be confused with that. And the next one we have is Russell's bodies, and this is seen in multiple myeloma with Port's bodies and like different one. So this is one Russell's bodies, and it is a large spherical eosinophilic inclusions found inside plasma cells composed of IgEs, that is immunoglobulins. All right, so these bodies are made up of immunoglobulins in multiple myeloma case. And then in heart failure cells, you must have heard about this is seen in chronic pulmonary congestion due to left heart failure, and they look like this, and these are hemosiderin laden macrophages. All right, so siderophages, we also talk about siderophages is heart failure cells or macrophages in the lungs that have engulfed red blood cells and the iron-rich pigment hemosiderin. So that siderin causes siderophages. All right, so that is about heart failure cells. The next one we have is McCallum's plaques, and they are like typically seen in rheumatic endocarditis, and they look like this. This is a gross feature, not microscopic feature, so you should not be confused with that. So the microscopic feature will be having a different composure. All right, so McCallum's plaques, this is a thickened, wrinkled, map-like areas on the inner lining of the endocardium. You can see it in the attached figure right here of the left atrium in the heart. All right, it is seen in left atrium. So the next one we have is Mallory-Denk bodies, and this is seen in alcoholic liver disease. Looks like this, obviously, and these are cytoplasmic protein clumps found in liver cells or hepatocytes. And the 14th one we have is Councilman bodies, and these are also seen in viral hepatitis, also a liver disease, obviously. So the arrowed portion, you can see, pause the video and look at it. All right, so this is a microscopic eosinophilic globules that are found in liver cells that indicate cellular injury, specifically apoptosis. Apoptosis is a programmed cell death, and unlike necrosis, all right, so it can be due to necrosis also, but specifically it is due to apoptosis. So we have ground glass hepatocytes next. So it is seen in chronic hepatitis B virus infection. They look like that, just like ground glass. And the description for this is that liver cells that appear hazy and pale on a microscope slide due to accumulation of hepatitis B surface antigen. We have different antigens in hepatitis B: surface antigen, core antigen, all right? So in their endoplasmic reticulum, so these are seen like the HBsAg is accumulated in endoplasmic reticulum of the liver cells, all right? And that gives appearance of hazy and pale. So the 16th one we have is Rokitansky-Aschoff sinuses, very famous sinus, and this is seen in chronic cholecystitis, and they look like that. This is a it is a diverticula-like species, all right? So benign diverticula-like species in the muscular wall of the gallbladder caused by a hyperplasia and herniation of its inner lining. We all know what a herniation is, all right? So that is how Rokitansky-Aschoff sinuses look like. Then we have next ciliary bodies. This is seen in yolk sac tumor. This is my personal favorite bodies actually. Why is that? Because it is very easy to explain and to remember. You have three different structures in three different levels. In the center we have central blood vessel or capillary, all right? So it is a pericapillary structure, and like that blood vessel is surrounded by a tumor cells, and that tumor cells is surrounded by cystic space. You can see the white spaces in the figure right here, all right? So this is three-level structure. It consists of a central blood vessel surrounded by tumor cells that is within a cystic space. And while they can appear in about half of these tumors, they are considered pathognomic. And the 18th and last one for today's session is Michaelis-Gottman bodies. It is seen in malakoplakia, and this is how they look like. This is a magnification of 20 micrometer. And then these are actually mineralized layered structures found in the cytoplasm of histiocytes, all right? So the calcified remnants of undigested bacteria composed of iron and calcium deposits. So this is how Michaelis-Gottman bodies form. So that is all about today. Thank you for staying, and please subscribe for the next part. Thank you.